In this video lecture, we're going to discuss the cell membrane structure, all of its components, and what their functions are. So let's get started. So we pull away the curtain, and we'll begin with, just in general, what is the function of the cell membrane? This is something that we've mentioned numerous times before. If you look up top here, all right, you can see that we have UV dye has been used to stain the actual lipid bilayer here. So here's our cell membrane. And what we really thought of the cell membrane up until this point is it's just a lipid bilayer. So two layers of fat that you can see here with the tails that are in the middle. All right, but we're going to see in this lecture there's a lot more to it than just that. So our function is, again, it's a phospholipid bilayer. So it's a very specific type of lipid, a phospholipid that surrounds the cell, and it ultimately is responsible for regulating and controlling what comes in and what leaves, as well as supporting it and giving it protection. So ultimately the cell membrane is the outer boundary of the cell. Every cell has to have a cell membrane. It protects it, supports it, but it is also the gateway that decides what can enter and leave the cell ultimately. All right, there are going to be numerous components, as I mentioned, that we're going to go through. We're going to have 4 key components that we need to be able to know their function as well as visually identify them. And lastly, you can't be a cell unless you have a cell membrane, period. So anything that is living has a cell membrane, period. That's just the way it has to be. So again, ultimately, protecting and supporting the cell, as well as helping to control what enters and leaves. All right, and all the components that we're going to discuss is helping to contribute to this overall function. Now, the cell membrane, we'll become an art teacher here for a second. The cell membrane is like a piece of art. In particular, a mosaic, which is a picture that is made up of many different components that are all contributing to one goal. In this instance, a visually appeasing work of art. All right, I love this one here at the beach with the wave, just made out of loose tiles, broken up, put together in a particular condition. I even love how the sun here is glaring off the water. That's really cool. All right, the cell membrane is going to be the same thing. Lots of different components being put together to make the overall membrane. Now, what scientists call the cell membrane is actually the fluid mosaic model. Fluid meaning that every component of the membrane is in constant motion. They're not just sit like concrete, stuck in one spot. Every single thing you see down here, proteins, phospholipids, cholesterol, carbohydrates are actually what these are right here. They're all in constant motion, kind of like a floating in a sea of lipids. So, fluid, that everything is in motion. Mosaic, meaning that the cell membrane is made up of all these different components here. Phospholipids, cholesterol, proteins, and carbohydrates. Now, for composition, all right, 4 key components. We're going to start with the largest component. What I mean by largest, not size-wise, but builds the bulk of the membrane. At least 90% of the membrane are what we call phospholipids, put together into this bilayer, 2 layers of fat. So the function of the phospholipids, besides being the main component, are to provide the flexibility to the membrane. So the membrane's got to be able to shrink, expand. All right, the phospholipids are ideal for that. And also they help, like I said, control what can enter and leave it. Remember that lipids are generally put together the same way. They're going to have a glycerol head over here with fatty acid tails attached. We focused on, in our macromolecule unit, triglycerides. This isn't any different except that I don't have a tri. I don't have a third fatty acid tail. Instead, I have a phosphate group added to the glycerol head, which is really contributing to the flexibility. All right, what we need to remember here is that each phospholipid, or each lipid in general, is not always going to be soluble in water, meaning that it will not dissolve, and that has to do with its structure. So for phospholipids, the glycerol head is a polar molecule. All right, so that means that it is going to have some slight positive and negative charges and is not afraid of water. It'll be attracted to water. This is why you always see the heads facing either the outside of a cell or the inside of the cell where there is actually water. The tails, on the other end, they are just carbon and hydrogen atoms bound together, and they're going to be sharing electrons equally. So those are nonpolar, which means they're going to be hydrophobic. So hydrophobic means you're afraid of water. This is why the tails are always facing aligned to the inside of the cell membrane, because they don't want anything to do with water. They're going to repel it, and water is going to repel them. So this is always the basic structure here of a cell membrane that's broken down into a lipid bilayer. The hydrophilic or polar glycerol heads facing outside or inside, and the hydrophobic nonpolar tails, fatty acid tails, will be on the inside of the membrane. The second component of a membrane are proteins. All right, as you can see over here, the proteins are in motion. They're floating around in this sea of lipids. All right, proteins are going to be either embedded within the membrane itself, or you'll find them attached to the inner or outside surface of the membrane. So if you draw your attention down here, here's a protein that's fully embedded within the membrane. It sticks out on both sides. And then these two here would be peripheral proteins, or proteins that are attached just to the lipid layer itself, either outside the cell or inside. They don't go all the way through, and they're not embedded fully into the lipid bilayer. Either type of protein is going to act as a transporter or receptor for the cell. Transporter meaning that they are going to allow certain things in or out of the cell itself. They're going to aid in that. They'll also act as receptors, meaning that they could recognize other similar cells that are nearby and communicate with them. So based on where the proteins attach themselves, they either fall into one of two categories. The first one here being an integral protein. So they are permanently attached, so you can see how they're embedded within the membrane all the way. And the other ones I showed you over here that are attached just to the inside or outside surface, we call them peripheral. Peripherals are always towards the exterior. Alright, so they're going to either be attached to the lipid bilayer, like you see, or sometimes they may actually be attached to an integral protein as well. It depends. The point is, you will not see them going through and penetrating the entire lipid bilayer. Alright, so both integral and peripheral proteins are going to aid in transportation. If something, for example, is too large to fit through the lipid bilayer, or you have a particle that is polar and would be repelled by these hydrophobic tails, that's when they're going to use these protein channels, where they can get through. If they're too big to squeeze through the lipid bilayer, they can go through this tunnel, like a tunnel through a mountain. Or if they are small enough to fit through the lipid bilayer, but they be repelled by the hydrophobic region, they're also going to make use of one of these tunnels to get in or out. The peripheral proteins will sometimes grab something, and then they'll feed it into the integral membrane protein. So maybe they're not directly the result of transport, but they are aiding in transport. Third out of four, we have what we call oligosaccharide chains. Fancy word for sugars or carbohydrates. So if you see the prefix glyco, that means you have a carbohydrate chain. There's going to be two types. We're going to have what we call glycoproteins and glycolipids. All right, both are going to serve the same function. It's just a matter of, are these glyco, the sugars, attached to a protein, or are the sugars attached to a lipid? So if you look over here in the diagram, you can see that here is a sugar chain, oligosaccharide chain, same thing, attached to a protein. So this whole thing here would be a glycoprotein. Or if they're attached to one of the little balloons with two tails, one of the phospholipids, the whole thing itself is a glycolipid. So for glycoproteins, again, sugars or carbohydrates are attached to one of the proteins, either integral or peripheral proteins. And their function is going to be for communication between cells. So they look like little cell phone towers, right, coming off the surface of the cell. All right, these are going to be used to actually touch and interact with other cells nearby, or they can receive chemical signals such as hormones from other cells that allow cells that are separate from one another to actually communicate with one another. And this is very important, especially in your organs. You want all the cells in an organ, like a kidney or a liver, to be working together in unison. And then for glycolipids, again, same overall goal and purpose. But again, we're just going to have sugars this time attached to a phospholipid rather than a protein. But ultimately, the function, once again, is for intracellular communication, communicating between two or more different cells nearby. And our last major component here, cholesterol. Again, here's that overall structure of cholesterol we learned about in our macromolecule unit. Again, cholesterol is a lipid, but it falls into that steroid category because you have 4-ring structure with a fatty acid tail attached. Notice that the cholesterol molecules are kind of just sprinkled sporadically into the lipid bilayer, and they're actually embedded within it right next to the fatty acid tail. So the cholesterol, again, is nonpolar. It doesn't dissolve in water. So that makes sense why it's right next to the fatty acid tails rather than sticking out here. Alright, so the purpose of the cholesterol is to add strength and support or stability to the cell membrane. Also, it decreases its permeability. All that means is it slows down the ability of certain molecules to move in or out of the lipid bilayer. Alright, if a cell did not have cholesterol, such as plant cells or fungal cells, they're going to need to have a cell wall for added strength. Alright, what these cholesterol molecules are doing, they're just keeping the phospholipids from flexing too much in any which direction. Alright, you do not want your lipid bilayer to completely collapse and destroy all the organelles inside the cell. You also don't want them to expand to the point where the cell membrane could burst. So it puts a limit on how flexible the phospholipids can actually be. But on top of that, cholesterol is going to help to control the fluidity. So think of it like how liquidy the membrane can get. So the cholesterol molecules that are mixed in the membrane are going to be attached to the fatty acid tails. And what they'll do is, under high temperatures, they actually stiffen the membrane. As with anything in high temperatures, things start to move faster. If you put butter on a skillet, it starts to melt and become liquid. We don't want that to happen to our cell membrane. So in high heat, these phospholipids will try to move further away from each other, but the cholesterol doesn't let them do that. It kind of holds on to each one on each side, so all these membrane lipids can't move too far apart, causing the lipid bilayer to burst. Or, in lower temperatures, think about how most things become solid when they freeze, right? It's by getting closer together and becoming more dense, except for water that we learned is the opposite. So, in a very cold environment, these phospholipids would try to get closer to one another. Well, the cholesterol being in between doesn't let them do that, so your cell membranes prevents them from freezing. Now, if it got cold enough, obviously they could freeze, but they can still resist temperatures below the standard freezing point, 32 degrees Fahrenheit, and your membranes won't freeze. That's why your skin doesn't shatter when you go outside if it's below 32, because the fats and the membranes making up your skin, your epidermis, aren't freezing because cholesterol is prohibiting them from doing so. Alright, and lastly, when we look at this, here's our mosaic. Alright, this is our lipid bilayer, our cell membrane with all the different components. We want to be able to visually identify them. Cholesterol looks like this, sporadically mixed in. The phospholipids are the little balloons with 2 tails making up the lipid bilayer. Proteins used in transportation, again, the integral proteins go all the way through the lipid bilayer, while the peripheral ones are either attached to an integral membrane protein or perhaps just the lipid bilayer surface, either inside or out. And then we have the glycoproteins and glycolipids. Again, glyco meaning a carbohydrate chain. If it's attached to a protein, either integral or peripheral, we call the whole thing a glycoprotein. Or if the carbohydrate or glyco chain is attached to a phospholipid, see the little 2 tails there, the whole thing itself is then a glycolipid. Again, both are used in intracellular communication. So ultimately, what I want you to take away from this is you can visually identify all these components, as well as tell me ultimately what their function is for the cell membrane. Thank you very much.